Once again, a very good morning and a warm welcome to the first scientific session of the webinar. To chair the session, we have two very eminent scientists of repute, Dr. Nitish Mahapatra and Dr. Priya Srinivas. Dr. Nitish Mahapatra is a professor in the Department of Biotechnology at the Indian Institute of Technology, Madras. His research focuses on the molecular basis of cardiovascular and metabolic diseases. Studies from his laboratory have identified several human genetic variants that significantly enhance risk for cardiovascular and metabolic diseases. The incisive studies have revealed mechanisms at the transcriptional, post-transcriptional, and post-translational levels, and the findings have implications for the clinical management of cardiometabolic disorders. Professor Nitish has published over 70 research articles in major international journals. He is a recipient of a number of awards and honors for his scientific contributions. He is an elected fellow of the National Academy of Sciences India and the American Heart Association. It's our pleasure to invite Dr. Nitish Mahapatra to chair this first session. Dr. Nitish Mahapatra, welcome, sir.
Thank you. Thank you so much for the kind introduction. And at the outset, I would like to also thank the organizers, especially Dr. Sivakumar, for inviting me to associate with this meeting. And I welcome my co-chair, Dr. Priya Srinivas from RGCB. So, would you like to introduce Dr. Priya before we proceed?
Yes, sir. Thank you, sir.
Okay.
Dr. Priya Srinivas is a senior scientist at the National Center for Biotechnology, Toronto. She is a distinguished cancer research scientist with about 3 decades of research experience on treatment-effective breast and ovarian cancer. Dr. Priya has been a recipient of prestigious fellowship to work in reputed institutions such as the National Cancer Institute and Mayo Clinic in the US. In 2013, she received the Prema Vahi Award of the Indian Council of Medical Research. Recently, she was awarded the 2022 ICMR DHR International Fellowship for Senior Scientists. Dr. Priya has about 50 international peer-reviewed publications in high-impact journals. We are delighted to invite Dr. Priya Srinivas to join Dr. Nitish Mahapatra as chairperson for this session. Dr. Priya Srinivas, welcome, ma'am. Over to you, chairpersons.
Thank you. Thank you for the nice introduction. Dr. Nitish, I think we can start the session right away. Yeah.
Sure. Okay, welcome everyone to this first session of the technical talks, this webinar. So our first speaker is Dr. Alan Sam Titus from the Department of Cellular and Molecular Medicine, Rutgers University, USA. He's a postdoctoral research fellow working over there, associated with Dr. Junichi Sadoshima, a well-renowned scientist in this area. So Dr. Allen did his master's in biology by research at the TIFR, focusing on the role of Citrulline-4 in regulating energy homeostasis and nuclear mitochondrial communication. Then he did his PhD at Sri Chitra Tirunal Institute of Medical Sciences and Technology at Trivandrum, where he worked in the area of cell and molecular cardiology, particularly the cardiac fibroblasts, understanding the molecular basis of the cardiac fibrosis. So I happen to know about his work closely because I served as the examiner of his PhD thesis. So it's a really great opportunity for me again to meet Alan over here in this forum. And then he is also an expert in the area of development of knockout and knock-in mouse models, wherein he works in the regulation of SARCA pumps under oxidative stress conditions in the heart. So Alan has published actually a lot of papers, including in the Journal of Clinical Investigations, Journal of Biological Chemistry, and American Journal of Physiology. So it's a great forum for us to hear to Alan regarding the knockout mouse models that he will be talking about today. So over to you, Alan.
Yes, thank you, sir, for the kind introduction. Let me see. I'll just start my presentation. Yeah, so without much delay, I'll just start with my topic. So today, or for the seminar, I'm focusing mainly on the advances in the generation of knockout mouse model of human diseases. So as we all know, just to give a brief introduction of what disease is, and then how we can tackle or molecular level how a disease can be studied. So a disease which can be defined as an impairment of a normal physiological function of all or a part of an organism caused by external factors, which includes pathogens, or intrinsic dysfunction, which could be genetic or other epigenetic modifications. As you see, I've shown here the strata, various levels of like an organism where you can study a disease physiology. And you can see that these strata are interrelated from the molecular level to an organism organelle level, cellular, to a higher an organism level, to a meta-organism level where the host pathogen interaction is also involved. So looking to the pathogenesis, when we try to understand the pathophysiology of a disease, all the mechanisms can be like pointed down or traced down to the molecular level, and it can be described from a molecular biology point of view. So in that respect, the proteins, they are the workhorses of the cell, and they perform a lot of function in the cell, which includes not only it forms a major structure and mechanical part of the cell. It performs various enzymatic reactions, metabolic activities, hormones, immune mediators. It is involved in pH balance, transport function, and also various channels and protein pumps. It is involved. So therefore, it is important to see or to understand, or as we all know, the proteins are coded by an intrinsic mechanism which involves DNA to RNA to protein, which is the central dogma of molecular biology. And this is not just a linear pathway, it is also a bidirectional from the RNA to DNA. It can reverse transcript and can modify the epigenetically modify the DNA and its regulatory transcriptional mechanisms. Also, the RNA can be modified or regulated at the level of splicing machinery. And not only that, RNA not only code for protein, there are also non-coding RNAs, which has also a lot of regulatory function in the cell. So therefore, not only proteins are the important factors which can be like focused as a part of disease metabolism or disease phenomenon. We can also look at various other factors which contribute to finally a protein which can synthesize into a protein phenotype, which involves epigenetic modification and regulation, the splicing machinery, as you can see, and also the non-coding RNAs which are involved in these processes, which regulates the expression of various other RNA, which ultimately lead to the expression of protein. So since for our topic today, we are looking at models, how to develop mouse models, it is important to discuss why mouse is an important model for studying human disease. We know that mice harbor most of the human biological process, since humans and mice share 99% of same genes. They are also important resource to study the underlying pathophysiology at an organism level, and also it is important or it provides a strategy for treatment or development of therapies for the treatment of disease. So why we use a mouse as a model is because as we all know, humans and mice share like almost 99% of same genes, and it is also an important resource to study an organismal phenotype for the development of disease. And basically, the laboratory strain of mice which are used for these studies are restricted from genetic and biological variables because they are inbreed strains of animals. So coming to the first, the main topic, the gene inactivation by knockout. So the knockout mice in which a specific gene is disrupted using various techniques as shown here, which can be a homologous recombination using a Kree-Lock-Spee system, Sinkfinger Nucleuses, talons, and the most recent one, which is CRISPR Cas9. There are also others which I have not included here, like the flip-apart FRT system, which for the sake of time I have not included here, but there are also other methods where you can introduce gene inactivation. So coming to the first one, which is the homologous recombination method, which was the first method employed for the generation of knockout mice. And for that, three eminent scientists, Mario Capecchi, Oliver Smithies, and Martin Evans, were awarded the Nobel Prize in Physiology or Medicine in the year 2007. So the first two scientists, as Mario Capecchi and Oliver Smithies' work, were mostly involved in the targeting of the gene for disruption. And Martin Evans' work mainly contributed to the cultivation of embryonic stem cells, which is very much important for the production of the mouse. So the first half is the generation of the recombinant DNA, and the second half is the incorporation of this recombinant DNA into the embryonic stem cells, which can be used to develop the mouse. So that is how this work got divided into these three people. So to explain more detail how what are the steps involved in the generation of knockout mouse, we can start with a DNA which is cloned to target to a particular gene of interest where we want to disrupt the gene. You need to first clone a DNA segment which contains the gene of interest, which is also associated with a thymidine kinase gene, which can be used as a marker. Like when after the complete process, you can see check for the presence or the effect of the thymidine kinase gene, and then select the non-recombinants using this particular marker. Along with that, to disrupt the particular gene of interest, as you can see, so here you have a target gene of interest which you plan to insertional inactivate the gene in the organism, which is also associated with the thymidine kinase. And then the neomycin resistance cassette is targeted within the target gene with flaps containing the regions of homology where you want to insert the gene of interest. And this construct is electroporated and transfected into the embryonic stem cell from a mouse which is brown coat in color. And there are three possibilities of recombination or three possibilities that happen. So the first possibility is proper insertional inactivation, where the recombination happens between the homologous regions of the gene, targeted gene, and you get insertion of the neomycin cassette into the gene of interest, where the neomycin cassette get incorporated. And in this scenario, since the neomycin cassette is inserted, you can select the embryonic stem cell which has neomycin resistance, and then collect them for further process. And the others which have a non-specific or no recombination can be ruled out with the absence of the thymidine. With the absence of no insertion, mean there is no insertion of the neomycin cassette, whereas In the non-random insertion, where you don't want where you're not getting a proper recombination, you can rule out the cells by giving the or providing the cells with GAN cyclover, which will be like metabolized by the thymidine kinase gene, and it becomes lethal for the cells to survive, and you get to only get the neomycin cassette resistant embryonic stem cells to be alive in the condition. And this post-selection, the embryonic stem cells are with the knockout allele are then implanted into the embryo of a surrogate female black mouse, and this is transferred to a surrogate mother, and the resulting mouse will be a chimeric mouse because the embryo which is coming from a black mouse, and also the original mouse from where the gene insertion was done, that is was having a black coat, you get chimeric mice, and this is back-crossed with the black wild-type mouse to result in the F1 progeny, with the brown coat ones will be the ones which are heterozygous for the gene insertion, and this heterozygotes can be mated with their siblings to get a proper homozygous gene knockout for the gene of interest. Am I with you?
Yes, yes.
So there are various gene disruption strategy. One is where you insert the homologous regions with the neomycin resistance cassette, and that gets replaced in and losing exon 2, where you disrupt the gene. And another strategy wherein you don't want to disrupt a complete set of a region from a gene. You just want to only introduce a gene point mutation where a particular amino acid residue only from a protein needs to be changed to another amino acid. In that kind of strategy, how it is the gene transfection strategy is developed as shown here, where here you can see that the exon with the insertional or with the point mutation or the subtle variation is cloned along with the neomycin resistance cassette, and with the neomycin cassette will be introduced with the flip the flocks sites. So these black arrows are flocks sites. I'll come to in a bit what the flocks method of recombination is. So this is done in such a way that the neomycin cassette is like targeted between the two flock sites, such that later on, after the first set of recombination, post neomycin cassette resistance selection, you can add a Cre recombinase transfection to remove out or lariate out the neomycin cassette to insert the exon 1 with the point mutation. So such that we don't actually lose the total exon 1, except that it is replaced with new exon 1 with the point mutation. So the problem with the whole body knockout, so the whatever strategy which I've discussed so far is where it is a full body or whole body gene inactivation or gene disruption is happening. So in some cases, when you do a whole body knockout, these genes may be like very much important for embryonic development, or they are very much vital for the life supporting activity of the cell or the organism. So in that case, when you actually do a complete gene disruption, this it becomes an embryonic lethal, and it will not develop into an adult organism, and you cannot study the function of the gene. So in such cases, the again more recent methods where you have conditional activation of the inactivation of the gene system can be done. wherein, as I mentioned earlier, also the Cre-LoxP system comes in handy. So the Cre-Lox enzyme, the Cre enzyme, and the original LoxP sites were actually derived from the bacteriophage P1 system, where this was as a part of the bacteriophage replication. The LoxP, the region flanked within the LoxP sites is like lariated out and then taken out for the production of more viral particles. So this basic knowledge was further used to develop a system where you can actually use this for recombination and addition or removal of the gene of specific interest we have. And therefore, investigators are often interested in examining the effect of knockout mutation in a particular tissue during specific stage in the development, just because we have a system where you can activate the LoxP system only when the Cre is added into the cell. So I'll come to the explanation. So here in the LoxP site, the strategy is that, as earlier shown, also the region where you want to remove or add the disruption, the neomycin cassette, which is the selection marker, is flanked within the LoxP sites. And this, after the first set of integration and selection with neomycin cassette, you can extraneously add or put the recombinase enzyme into the system by externally driving it through transfection, or the Cre can be put under the specific tissue-specific Cre expression, and can lead to the expression of Cre and lead to the existence of the neomycin cassette for the production of various 3 different recombined recombination. So there are 3 possibility of after adding the Cre recombination. One is you will since there are 3 regions of floxing, the exon 3 exon 2 along with the neomycin cassette is lost or lariated out, and only the one flox site with the exon 3 is made. Another is the properly recombined chromosome, where we want to remove only the neomycin cassette region and want to introduce the flox region on both sides of the exon 2. And the other one is where the exon 2 is removed and the neomycin cassette along with the exon 3 is inserted like that. So this can be so these are the 3 different embryonic stem cell population that we get out of the transfection with the Cre recombinase. And from these population, we can do PCR and identify which of the clonal population is actually having the insertion, where we design primers that will amplify only the regions with specific gene of interest. And that is how you identify a particular recombined clonal population, and that you can the same procedure as shown above. This embryonic stem cell can be developed into a knockout mouse with a conditional Cre expression. So the LoxP site, which was introduced onto the exon 2, the mouse is a separate one. So here the mouse, the gene is functional, it is normal, but we can this gene expression can be disrupted only when the Cre expression is driven using a cell type specific promoter. So for this, the LoxP mouse which is developed can be crossed with a Cre mouse, which is where the Cre is driven under the cell type specific promoter. And the progeny resulting from these cross will have a tissue-specific Cre expression. For example, in heart, we use the alpha MHC myosin heavy chain promoter to drive the Cre expression. And only in the heart tissue you will have Cre expression, and the Cre results in this heart tissue, the recombination and the excision of the exon 2, leading into gene function disruption. So this is a kind of a convenient model where you can specifically disrupt the gene function only later on in the adult stage, or in a tissue specific manner. Further, there are again more modifications come. Okay, before that, there is a directionality for the log speed existence system. So the log, yes.
Sorry to
Yes.
interrupt you. So I think we lost some time. So in order to kind of keep this schedule, we need to kind of finish up in. So maybe in another 5-6 minutes, can you wind up?
Yes, possible. Yes, that's true. That's
It's
possible.
right.
Yeah.
Okay.
Yeah.
Sorry, but you have.
That's okay. Yeah. Yeah. So the next method is like where you have an inducible knockout system where under a tissue specific expression, you have the tetracycline activator protein, which can be made only in the presence of doxycycline to drive the expression of Cre, resulting in the removal of the gene of interest only in the presence of dox. And there is another mode which we also use in our lab, is the tamoxifen inducible system, where the Cre is modified in such a way that it is fused with an estrogen receptor, a modified estrogen receptor, such that this is not active in the normal condition. This will only become active in the presence of tamoxifen. The tamoxifen is a modified estrogen a molecule which will, when binds to this estrogen receptor, it becomes active and becomes translocated to the nucleus and leads in the LOX recombination and the gene excision only in the presence of tamoxifen. So, which is also an inducible system where the Cre is modified with the estrogen receptor molecule to drive the expression. Again, another more specific way of doing gene disruption is with made possible was with the Sinkfinger Nucleuses, which was developed during the early 2000s. So this is actually like the Sinkfinger nucleases, which are the DNA binding motifs, or the most of the transcription factors which has the loop helix structure, where the loop is actually stabilized by a zinc moiety. So the loop along with the helix, the loop along with the helix structure have a property where it can specifically bind to certain specific DNA sequences. And this Sinkfinger nucleases, when it is combined with the recession enzyme, which is FORK1, which has a capacity to make double-stranded DNA breaks, it can be used for the disruption of specific gene sequences where you want to like either disrupt the gene expression, or if you want to introduce a specific gene of interest in by homologous recombination, you can introduce that specified DNA at the sites of double-strand breaks to introduce that particular gene of interest. Where so one is method is by homologous recombination, you can disrupt the gene, or by homologous recombination when you add a gene of interest or something cassette which can be inserted in a specific manner. Then we have the another system which is Darlin, which is like developed from the Xanthomonas bacteria, the system which comprises of an invading domain and a DNA binding domain and nuclear localization domain and a transactivation domain. So the DNA binding domain is actually a protein which is an amino acid sequence which has a 33 to 34 long amino acid sequence, wherein the 12th and the 13th amino acid is responsible for binding to the DNA sequence specifically. So the amino acid combination at the 12th and the 13th residue of this domain specifies its specificity to a nucleotide, like as shown here in the tail code. So this is actually a repeat where you have the domain, the repeats come close by at the 12th and the 13th residue, and through that it can bind to specific region of DNA. So this, when conjugated with fork1 nuclease domain, can be used to specifically introduce double-stranded breaks, or this DNA binding domain with the tailed repeat domains, if conjugated with an activator, can act as a gene activation system. Also, if this is conjugated with a repressor, can downregulate the gene expression. And finally, the CRISPR-Cas9 system, which is the most recent system, which is developed, which is developed by the two eminent scientists for the gene editing, which is Emanuela Carpentier and Jennifer Doudna. In the year of 2020, they received a Nobel Prize in Chemistry for their discovery and the use of the CRISPR-Cas9 system for gene editing. So in the CRISPR-Cas9 system, this is actually an endogenous function where the exogenous or foreign DNA can be degraded using the CRISPR-Cas9 system. It's a kind of immunologic response by bacteria towards foreign DNA. So this system was manipulated and used for gene editing purpose. So basically, what we have is that there are various repeats of DNA which contains a portion of the target DNA from the foreign DNA which the host is planning to target and destroy, and that is found in the CRISPR locus. And the CRISPR locus, after transcription, gives to a primary transcript with the CRISPR RNA sequence interspersed between the gene of targeting. So this, along with the tracer RNA, combines to form the CRISPR RNA system, which is identified by the Cas9 enzyme, which is an endonuclease, and the region of homology between the target sequence and the foreign DNA is matched, and the endonucleases within the Cas9 enzyme cleaves the DNA to provide the sites of recombination. So in the natural CRISPR pathway, this was supposed to be to evade foreign DNA, but then this Cas9 system has been engineered in such a way that this can be used as a tool to modify the DNA of our interest. So where the guide RNA, we can actually, instead of the DNA, foreign DNA recognition region, we can engineer the target sequence where we want to target the guide RNA. And this expression system, along with the Cas9 system, can be overexpressed in the embryonic stem cells, leading to the knockdown or the breakdown of the double-stranded DNA at the region of the target specified. So with the region specification, you can have the deletion or insertion where at the region of the double-strand break, it can insert or cause gene frame shift can occur. Also, if you introduce a donor DNA with a specific sequence where you want to insert at the site of double-strand breaks, that can be also possible, and that is possible through the homology directed repair. Further, you can also target the Cas9 system at two target regions where you can remove out or excise out a particular long segment of DNA. And again, you can use the homology directed repair to excise and add in the gene of interest for the same purpose. This also has been modified. The Cas9 enzyme has been modified in recently into various other mechanism where we don't inactivate the endonuclease system and add on to the Cas9 system an activator or a repressor or a GFP for the visualization of the cells where this is expressed. So in summary, the gene expression is a result of closely regulated processes that happen sequentially within a cell from DNA to RNA to protein. And the mutation in the genes can be reflected in proteins, and which leads to a pathological condition in the organism. And the tools which are required for the study of genes have been very instrumental in studying various pathophysiology of various diseases. And we owe a lot to these inventors for these techniques that have enabled the biomedical research to make rapid strides and understand human disease on an unprecedented scale. So these are the most of the references from where I've taken the images and the literature. And thank you for listening, and I'll take any questions.
Thank you. Thank you so much, Dr. Allen, for this excellent update on the techniques that are used for knockout mice generation. So we can take one or two quick questions from the audience, since we are going a bit delayed. Any quick questions from the audience?
Can I ask one question?
Yes.
Sure.
Yeah. So it was a very nice detailed presentation on this various models that is possible. But when we use TALEN or Greylock or Sinkfinger Nucleuses or even CRISPR, how are we going to use this for the treatment of human diseases? Could you please elaborate a little bit on that aspect? Because these are all study models, mouse. When you come to the human diseases, would you please elaborate a little bit on that aspect?
Okay. So basically, these are models where tools which we use to study the disruption of gene and then study what happens when this particular gene is disrupted. But in case of gene therapy for human application, where in a particular situation where you identify that the gene is disrupted in that patient, and for expression of particular or for the therapy of that particular gene, this techniques can be used where, for example, you can adeno-associated viruses for gene therapy have been used, where you can replace or insert the gene of interest which is properly functioning into the organism, into a human. So even in my study currently, which I'm doing, the SERCA protein, which is non-functional in oxidative stress, and there are various studies for gene therapy using adeno-associated viruses to correct the knockout or the disrupted gene expression in failing heart, and they have shown that the expression of normal functioning counterparts of the SERCA protein was able to rescue the disease phenotype. So yeah, there are this is just one example which I'm showing. So there are various vectors which can be used for the expression of the targeting genes. For example, any mutations are there, you can replace the mutated gene by targeting using various vectors available from the various systems.
Okay, thank you. So I think we'll move back ahead because we are almost 15 minutes
Yeah,
delayed.
thank you so much.
So thank you, Alan, for joining us in this session, and all the best in your future work.
Thank you, sir. Thank you.
Okay.
Yeah.
Okay. So